Great. Well, thanks to the organizers for the chance to contribute to this innovative initiative. My name is George Bassel. I'm based at the University of Warwick.
George, you got muted, I think.
Sorry, have I been muted?
Nope, he's talking.
The volume, yeah.
Can you hear me okay?
Yes, you're good.
Okay, apologies for that. So yes, we've heard quite a bit about already about the scales in the organization of biological systems. So I won't go into too much detail with this, but they can be viewed as a hierarchy, starting with the genome and ending in the phenotype. And then what you see is a progressive series of scales through which information flows. So the genome has genes expressed, which encode protein, which in turn bind DNA and other molecules, and other proteins as well. So what we're seeing here is this drive in an increase in complexity as you traverse through these scales. And there's a big jump going from the molecular scale to the organizations, they have organelles within cells and cells within tissues, and then in turn to the phenotype. So I've highlighted those for a reason. I'll get to that in a moment. So what we can say then is that the phenotype is emerging from genotype via the traversing of information across multiple scales. If we consider these various scales, we have an asymmetry in terms of the availability of data across each of these scales and the throughput with which the data can be generated. And this is really just down to technology at this stage. So we can sequence genomes quite readily, profile gene expression. Phenotyping is coming online very much so at the moment. But it's these mesoscales, for example, the organization of organelles within cells or cells that make up tissues, that we have the least amount of data. And it's this latter point that I want to speak about today. So in my lab, I've had this very talented postdoc named Salva Duran and Breda. And what Salva did was to create a 3D digital tissue atlas of Arabidopsis. So what he's done is done whole mount 3D imaging and 3D image analysis of the various tissues that are represented here in this schematic. And what he's done is a 3D segmentation of each and every cell within each of these tissues and annotated them by cell type. And what we've done, therefore, is digitally capture both the 3D shapes of the cells. So these are the various cell types that are making up the major tissues of Arabidopsis. But importantly, what we also have is the connectivity of cells which make up these tissues. So we have cell connectivity networks. So in these networks, the nodes are representing cells and the gray lines, their physical associations. So in panel A there, this is actually an Arabidopsis root, even though it doesn't really look like it. And these are all the various cells in the root and their physical associations. So what we're able to then do is the fact that we're able to take these images and discretize the organization of cells is to quantify cell organization using network science. And we've talked about this in the past. I won't get into it right now. But for the purpose of the plant cell atlas, these also represent the templates, these multicellular templates upon which all the genetic and biochemical programs that are being executed in a mature plant tissue are occurring upon. So it's not just the abstraction of data into networks of tissues that we've seen in the last couple of years. We have data structures represented as networks for all the scales that I mentioned previously. So starting from, say, gene on the left here on my screen, gene regulatory networks, protein-protein interaction networks. We haven't done this yet in plants, but in animals, they certainly made cell organization networks where the nodes are representing different organelles, and the edges, the frequency with which they physically associate. Tissue organization, as I mentioned, and as well phenotyping. You can abstract the architecture of a plant into a network as well. So we have a harmonized data type now, which can be integrated because these are all networks, they're all nodes and edges. And the integration which occurs can be done through a newly developed mathematical structure called multiplex and multi-slice networks. So these are networks of networks whereby each slice represents a layer or a scale. So in the example on the right here, you have a protein layer and below it a gene expression layer. And the edges are transposed across scales. So these are equivalent nodes represented on each of the layers. And it's these inter-edge nodes that are representing the transfer of information across the scale. So these in this instance would be protein DNA interactions. So this is important because you're able to integrate data types across the scales in a quantitative manner and in a discrete manner. And we proposed this together with Salva in a Trends in Plant Science paper back in 2017. And here you can see the data availability, asymmetry, and the various scales ranging up to ecological networks. So we have a framework here where we can integrate diverse data types and capture and quantify emergent phenomena. Recently, we published a paper just last year in Cell Systems where we did this in the Schudepo meristem. So if you'd like to read about how this can be applied to an actual example, please go ahead and read this paper. Happy to answer any questions. And I'd like to thank the people who did the work, Salva Durand-Breda in particular, Matt Jackson, PhD student, Hao Shu, a postdoc, and my funding. So thank you very much.